All right. Welcome everyone to our session. We're so glad that you are here with us today. Um, so we're going to get started. Before we get started, we do have a few housekeeping items for you. First, closed captioning can be turned on by pressing the closed captioning icon just above the video. If you run into any technical issues or need accommodations, please email all of us, Science Day, at pyxispartners.co. To ask any questions, we do ask that you use the Q&A option at the top of your screen. You can type your question and submit it. Questions submitted through the chat box will not be monitored. So we do ask that you use the Q&A box. So now we'll get started. All presenters will focus on turning research into better care. And I am delighted to introduce our panelists. Please join me in welcoming Cole Bird, a third-year medical student at the University of Kansas School of Medicine, John Shiyun, a research assistant at Stanford University, Chris Eisbouts, an industry researcher at NCITRO, and Jorge Garcia Diaz, a medical student at San Juan Baptista School of Medicine. During this session, our speakers will present. We'll have two of them present, and then we will give Q&As for those speakers. We'll then turn to our last two speakers, and after that, we'll wrap up with Q&A for those final speakers. If you have any questions, again, please put them in the Q&A panel, and we will get to them during the Q&A. So let's begin today with Cole.
Good afternoon, everyone. I'll take a second to say hi and say I'm very excited to be talking about my team's research entitled Wearable Sleep Monitoring Reveals Improved Sleep Architecture After Sinus Surgery. You can progress to the next slide. Chronic rhinosinusitis, or CRS, is a common inflammatory condition that affects far more than the sinuses alone. Many patients experience poor sleep, fatigue, reduced daily functioning, and lower overall quality of life. This matters because sleep is essential for cognitive performance, mood, immune function, and recovery, so chronic disruption can affect nearly every part of health. This is especially important because millions of adults live with CRS, and many ultimately undergo endoscopic sinus surgery, or ESS, when symptoms persist despite medical therapy. Prior studies show that patients often report sleeping better after surgery, but an important question remains. Is sleep actually improving physiologically, or are we just capturing subjective improvement? That gap motivated this study. We wanted to move beyond patient-reported outcomes and determine whether sinus surgery is associated with measurable changes in sleep architecture, particularly restorative stages like deep and REM sleep. Put simply, we wanted to know whether surgery changes how patients truly sleep at home, not just how they say they feel. Next slide. To answer that question, we used the ALS research program, the large national data set, and we used both the EHR data as well as the wearable device data derived from Fitbit sleep measures. This allows us to examine sleep continuously and objectively in patients' real-world environments. We identified adults with chronic rhinosinusitis and compared to those who underwent endoscopic sinus surgery and matched CRS patients who did not. Then we evaluated sleep patterns before and after an index date, defined as either the surgery date for the surgical group or a matched interval for the controls. Our primary sleep outcomes were total sleep time, deep sleep, and REM sleep. Total sleep time reflected the sleep quantity, while deep and REM captured more restorative aspects of sleep physiology. What makes this study especially novel is the use of the wearable device to measure sleep continuously over time. Rather than relying on a single sleep study or survey, we're capturing how patients actually sleep at home, night after night. This makes this approach more scalable and more patient-centered for evaluating outcomes after sleep. Next slide. Our findings show a clear and important pattern. At baseline, surgical patients started off with worse sleep physiology than non-surgical controls. In particular, they had lower REM sleep and lower deep sleep, suggesting these patients who ultimately underwent surgery may have had more impaired restorative sleep to begin with. After surgery, REM sleep improved significantly. Surgical patients experienced a substantial increase in REM sleep, and that improvement was greater than what we observed in the non-surgical control group. This difference finding suggests that the observed improvement was not simply due to time alone, but may be associated with the surgical intervention. Deep sleep also showed a smaller directional improvement. While that signal was not as strong as REM sleep, it still suggested a trend towards better restorative sleep after surgery. More importantly, sleep time itself did not meaningfully change. This tells us something very interesting. The benefit of surgery may not be that patients are sleeping longer, but that they're sleeping better. So overall, the takeaway from this slide is that ESS appears to be associated with improvement in sleep quality rather than sleep quantity. This surgery may be helping patients spend more of their time in restorative stages, which could translate to the better daytime functioning, recovery, and overall sleep time. Why it matters, the final slide. These findings matter for several reasons. First, they suggest that symptom improvement after sinus surgery may reflect true physiological change, not just a perceived one. That strengthens the case that ESS may influence whole body health rather than just nasal symptoms. Second, this work provides objective evidence that complements traditional patient-reported outcomes. Those measures remain essential, but wearable data adds this continuous, trackable view of recovery that is less dependent on recall or one-time assessment. Going forward, the next steps are to expand to larger prospective cohorts, integrate clinical outcomes such as the SNOT-22, and incorporate additional wearable metrics like heart rate variability and activity. Ultimately, our goal is to develop digital biomarkers that can better guide patient selection and monitor recovery over time. Thank you.
Thank you, Cole. Next will be John.
Thanks, Danielle. So I'm going to be talking about our lab's work, identifying glaucoma risk using systemic EHR data, using All of Us, and then externally validating it on a local clinical health system. Next slide. So the main condition that we're looking at here is glaucoma, which is one of the leading causes of blindness. And so glaucoma, it can be hard to detect early, but early detection and treatment can really help to slow the progression and lead to better patient outcomes. Unfortunately, glaucoma is only diagnosed a lot of the times after vision loss has already gotten a lot worse, irreversibly so. And a lot of the commonly used screening techniques to try to identify high-risk glaucoma patients require specialized ophthalmic equipment that might not be accessible everywhere. So in particularly, we're trying to target these individuals who might be at risk of glaucoma, but aren't being seen by eye specialists or getting the proper ophthalmic care. Next slide. So the main question we're trying to answer here is whether we can use routine non-ophthalmic clinical data to try to identify patients that are at high risk of glaucoma. And then can we also extend this model to be used in different settings, like going from All of Us to a local hospital system? So our approach here is we developed a glaucoma risk assessment model using systemic EHR, using All of Us, and then we tried adapting the trained model to our local Stanford data to assess whether the model was portable and whether it would be able to generalize well to other populations. Next slide. So our model, we found that the deep learning model achieved pretty strong performance for identifying glaucoma risk. We had an area under the curve around 0.86, and this outperformed other machine learning methods like XGBoost, et cetera, in the first plot. And then after we fine-tuned to the Stanford specific cohort, we found that the model still performed pretty well with an AROC of around 0.88, demonstrating that there was good transferability to the local population as well. And then we also computed sort of the correlation and calibration of the model predicted risk with other clinical quantities, such as like intraocular pressure and cup to disc ratio, where their anomalous values are also closely associated with glaucoma. And we found that the model predicted risk was really highly correlated with those results as well. Next slide. So yeah, why this matters. So we found that using the national All of Us data and the data from the participants to help generate the model helped make the models very robust because it used diverse national data. And we found that using this could help support earlier identification and treatment of glaucoma. So this is both beneficial to us as researchers to be able to work with the data, and also should help improve care down the line by helping people find earlier care. And so we found that the results were also transferable across different health systems to our local health system, advancing sort of the goal of moving from just our research to real-world care impact. And then finally, we think that we developed an innovative EHR-based system that did not use specialty-specific health information to try to predict a specialty-specific disease that just uses routine data that is sort of available in all health systems. Yeah, that's going to be it. Thank you so much.
Thank you. Now we will go into Q&A. So the first question that we're going to ask is, tell us a little bit more about the motivations for your research. Both of your research are very inspiring. I think it really does demonstrate how research can translate into action. And so I'd love to know a little bit more about the background.
I'm happy to begin. This really came out of like hearing patients report, sleep is oftentimes one of the big issues in chronic sinusitis. Your sinuses are just inflamed and you just are fatigued all the time. And so it's, one of our big priorities in how we educate patients with CRS. But there was no data backing up, like, okay, what's happening? Because after surgery, they're like, hey, my sleep is better. And so this really came out of that paucity and trying to find ways to, test our hypothesis to generate some data so that we could then pursue prospective studies. So it was a really patient-driven question.
Yeah, in a similar way, a big motivation just came from having patients come in the clinic and already having developed severe glaucoma to the point where it was difficult to treat and it sort of improve their quality of life. So that we were trying to just dive into the root of the problem and just try to identify these patients in the first place so that hopefully we can try to improve their vision sooner rather than it gets too late.
That's great. And I love the ways in which you've taken something from like patient care into research and then back into patient care. And that's really fantastic. I think the impacts really speak for themselves of your study, but I'm wondering if there's something that you felt surprised by in your own research, and if there are like impacts that you would like to emphasize that like the importance of what you have found out.
I think one thing our team has gone back to is trust the patients even when the data is not there. Because, if some patients were coming in saying like, hey, like, I think that I want to pursue this surgery. I think it may help my sleep. And the only thing you did is turn to, a literature search and not come up with anything. You may believe like, hey, that's not a reason. I don't think it's strong enough cause. But in reality, it's just we're finding a gap. So trust your patients and do the due diligence.
I think the biggest surprise on our side was just probably how sort of good we were able to get our model to be, given that we were predicting a bunch of ophthalmic specific related quantities without using any ophthalmic data. It was just surprising to us that we could, for example, have a model that strongly predicted eye pressure without using any sort of eye related data, just like demographics, labs, and stuff like that. but yeah, just the sort of whole going from systemic data and being able to predict some specialty information.
I'm also wondering, you've done this work now. What does your work look like in the future? In what ways do you think all of us can continue to support the type of work that, or the trajectory of this research?
Yeah, my team is actually working on, we're actually a finalist for a secondary grant to start looking at the prospective cohorts. And so partnering with a wearable device company to look at this long term and then expand. And so it's really nice. We were able to test some of our hypotheses with the All of Us data, and now we're getting prospective. And now I'm headed back to the All of Us research workbench to see, are there other ways that we can shape our grant writing to really look at the scope? And so it's been an amazing resource and will continue to shape the funding mechanisms that I pursue, as well as the way that we can affordably test some of our early on questions. And so it is now integrated into my workflow for many projects.
Yeah, in a similar way, All of Us was like a amazing sort of starting point for us to start developing a model. In a similar way, we're also looking to sort of be able to apply our model prospectively to true screening populations. So far, everything we've done is mainly just been looking at retrospective health data. And then we're also looking to participate in a network study and apply our model across multiple institutions and just try to see how generalized it can be.
Well, we applaud you both, and thank you so much for your presentations. They're really, really insightful. So next, we're going to continue on with our next set of presentations. And so, Chris, if you would like to start.
Yeah, thanks a lot for having me. My name is Chris Eisbouts. I'm a statistical geneticist. And the work that I'm presenting today is about using All of Us in combination with other biobanks for genetic analysis. So we imputed or predicted a disease phenotype across two biobanks, namely UK Biobank. That's where we train a model in one biobank, UK Biobank. And then we generated a disease phenotype from that model in All of Us as a second biobank. And that helped us to show that this gene, IRS1, is linked to the development of a liver condition. I have one disclosure to make, which is that I am an employee and shareholder of InCitro. So my colleagues and I looked at MASLD, or metabolic dysfunction-associated liver disease, that is defined by liver fat accumulation in the presence of cardiometabolic dysfunction. It's initially symptom-free, but untreated MASLD can lead to fatigue, abdominal pain, and eventually jaundice. MASLD increases all-cause mortality and is also one of the leading causes of liver transplantation. It affects about 30% of the global population, and that number is growing. Even today, direct medical costs in the US exceed 100 billion USD. Therapeutic options are limited, so patients are advised to make lifestyle changes typically. And the treatment options like GLP-1s or Rezdiffra are expensive and not approved for all stages of disease. We want to develop additional therapeutics and therefore asked what genes are involved in the development of MASLD. Second, we're looking for robust results, so we wondered if our genetic evidence would replicate across biobanks. Our first goal was to quantify liver fat accumulation as a hallmark of MASLD. And a gold standard way of doing that is to measure the protein density fat fraction, or PDFF, by means of an MRI scan. In UK Biobank, in blue, this liver fat measure was available for a small fraction of participants, indicated by this small bar. In All of Us, we don't have it at all. So MRI and other imaging data are not currently available in All of Us. But what we do have in both biobanks is electronic health records and also biomarkers for liver fat. And we use these data to train a model that predicts MRI-derived liver fat in this subset of UK Biobank, and then deployed it in both biobanks to predict liver fat for hundreds of thousands of individuals. Then we use the broadly available genetic data to identify liver fat-associated genes. In UK Biobank, that yields the IRS1 gene. In All of Us, we hope to replicate this result. And we use these 22 shared predictors along the y-axis to train our model. including simple demographic information like age at the top, enzymes like ALP and ALT right below, and diagnosis like diabetes in the middle. For UK Biobank, the blue bars show these predictors are available for about 500,000 individuals, as marked on the x-axis. In all of us, the majority of them are available in 200,000 or more individuals. These predictors are generally similarly distributed across biobanks, so we observe no significant differences other than for cholesterol and LDL, which typically have higher values in the UK Biobank population. We benchmarked our liver fat predictions against 3 clinically relevant observations: mast cell diagnosis on the left, high PDFF in UK Biobank in the middle, and having steatosis of the liver in all of us on the right. In these RC plots, curves creeping towards the top left, so away from the diagonal, indicate better model performance. And performance in UK Biobank is slightly better than in all of us, but in both cases, predicted liver fat discriminates mast cell cases from controls. Finally, we performed genetic association testing using the predicted liver fat phenotype. In the European ancestry group, we adjusted for abdominal subcutaneous adipose tissue, which we imputed in the same way. And we then saw that in the all of us data, IRS1 was also linked to liver fat. In conclusion, we imputed imaging phenotypes absent in all of us, but relevant to mast cell, using predictors shared across all of us in UK Biobank. Our analysis of these imputed phenotypes strengthened the genetic evidence that implicates IRS1 as a causal gene in mast cell pathogenesis. And this highlights IRS1 as a promising therapeutic target for mast cell drug development that we're continuing to pursue. So thanks very much to all UK Biobank and also all of us participants for making this research possible. And thank you for listening. Happy to take questions after.
Thank you so much. I'll now go to Jorge.
Okay, so hi, my name is Jorge Garcia, and I'll be presenting our study on GLP-1 and metformin effects on cardiometabolic risk factor. So GLP-1 receptor agonist drug work with the management and weight control for patients with diabetes type 2. Also, GLP-1 receptor agonist drug improve insulin secretion and provide cardiovascular benefits in diabetes type 2. While GLP-1 receptor agonists show strong benefits in clinical trials, the real-world effectiveness may differ due to treatment delays and also disparities. Our study evaluates these differences in a large real-world cohort. So our objective is to compare GLP-1 therapy to gold standard in glucose and also lipids management in terms of cardiometabolic outcome and assess differences in response across diabetes type 2 population. So to address this objective, as we can see in research objective, we developed a retrospective study using the NIH all of us datasets, including over 6,000 adults with type 2 diabetes, and also a BMI over 25. Patients were divided into 7 treatment groups based on their medications regimen within 3 pharmacological agents, metformin, GLP-1, and also statin. And we focus on 3 outcomes. Number 1, the therapeutic inertia, which is the failure to intensify therapy on patients that met the requirement to increase the dose. Then number 2, time to glycemic control, which is the length of the time it took to achieve glycemic control in comparison with metformin. And also number 3, lipids effect analysis, when we compare the GLP-1 independent lipids effect in statin-naive patients taking metformin. And what we found are the following. When we go to figure 2 in results, we calculate therapeutic inertia. Here we can see in the red color, black patients, then the red bar color, the black patients, then the next one is white, then Hispanic, then female, and then male, and the blue one, it's overall cohort. And we stratified the data by race and sex, as I mentioned before, and the data suggests that Black patient has significantly higher rates than white patients, about 7% disparity that remain after adjustment. And also we found that sex-based difference was not statistically significant. As you can see on figure 3 for glycemic control, on the top of the figure, GLP-1 monotherapy in blue color was associated with a slower time to reach HbA1c less than 7% compared to metformin. And also the combination of metformin and GLP-1, which is the last bar in figure 2, therapy performed slightly better, but was not statistically compared to metformin. And when we go to table number 2, our data suggests that GLP-1 patients show a greater reduction in triglycerides, but the difference was not statistically significant when we compare the uses of the GLP-1. Then also the LDL outcomes were also not statistically different. So based on this finding, we can conclude that therapeutic inertia is unequally distributed between Black patients and white patients, and also that GLP-1 therapy may achieve glycemic control more slowly. Our project highlight that clinical outcomes depend not just on drug efficacy, but also on timely and equitable care. And we gratefully acknowledge all of us participants for the contribution, without whom this review would not have been possible. Thank you.
Thank you, Jorge. Next, we will have Princess.
Hi, good morning, everyone. My apologies for tuning in late. I did not realize I was participating as a listener. Hello, my name is Princess Sule. I'm a MPH candidate in Jackson State University. My focus is epidemiology. I don't see my presentation.
It should be visible now.
Yes, thank you very much. All right, so in my study, I'll be presenting the association between antidepressant use and poor glycemic control among adults with diabetes using All of Us research program. And so as we know, depression commonly co-occur with diabetes, and this can complicate disease management. And previous studies have suggested that antidepressant use is associated with glycemic outcomes, but the findings have been inconsistent. And to use, I can't see my, yes, thank you. And so for this study, I'm going to be examining the whether antidepressant use is associated with poor glycemic control among adults with type 2 diabetes. And for my primary objective,
Princess, sorry, we're having a technical difficulty on the background. Please just give us one moment.
Yes, ma'am.
You may proceed.
Thank you. So for my primary objective, I'm examining the association between antidepressant use and poor glycemic control, defining HbA1c greater than or equal to 8%. And secondary objective included describing the distribution of glycemic control in the study population and also quantifying antidepressant use among the study population. For the methods, I used a cross-sectional study design from the All of Us research data base controlled tier data set, and the study population was 6,015 adults. 6,015 adults was used, identified with electronic health data to identify if they have diabetes mellitus. And for the outcome, it was poor glycemic control, and for the exposure, I used antidepressant use for exposure. Logistic regression was also used to estimate the association, adjusting for age, sex, and race. The results among these 6,015 adults shows a 24.5% poor glycemic control among this study population, and a high prevalent use of antidepressants, 94.7% use. Initially, in the unadjusted analysis, antidepressant use was associated with higher odds for poor glycemic control, but after adjusting for age, sex, and race, the association was no longer significant, and the adjusted odds ratio was 0.99, and p-value was 0.877. So this suggests that the initial association was linked, likely influenced by underlying demographics and clinical factors. In conclusion, antidepressant use was not independently associated with poor glycemic control, and after the use of adjustment, and also these findings highlight the importance of accounting for patients' characteristics in observational research, and they also emphasize the needs for integrated management of diabetes and mental health to improve patient outcome. In future direction, I would like to use a longitudinal study design to check for causality. Hence, since with cross-sectional analysis, causality could not be checked for, and also explore more clinical variables and also explore more medication use. I would also like to acknowledge all of us participants for their contributions, and also thank Dr. Sandra McNeil of the Delta Research and Educational Foundation, and also my advisor. Any question?
Thank you so much. We'll now head into the Q&A portion of this section. So thank you again to these three presenters. Chris, I do have a specific question for you because you used all of us and also the UK Biobank. I'm just wondering what that process was like. I guess from a methodological point of view and actually technological, using it on the researcher workbench, and any lessons that you've learned or anything that you would want to share to people who would want to do something similar.
Yeah, that's a great question. Practically, I would say the transition was quite smooth. So I'd encourage other people in the audience or other researchers who are currently using UK Biobank to definitely try and switch over to using all of us as well. I would say what's really nice about all of us, especially for the type of genetic analyses that we're showcasing, is that you have way more diversity among your participants. Whereas UK Biobank, for example, essentially your entire sample size there is European ancestry individuals. In all of us, that's not the case. And so that offers many opportunities for interesting genetics research. From a computational point of view, you could see in some of these slides that I presented is that we were harmonizing predictors for liver fat. And in all of us, these are presented in a very heterogeneous way, where you have to change the units to make them compatible. In UK Biobank, I would say typically that's done for you. So I think there's also a lot of interesting work that can be done on the all of us side to sort of help people make these predictive models. Yeah, good question.
Thank you for that answer. Kind of in a similar vein, but definitely different in terms of methods. I think all of your work kind of goes toward thinking about differences in populations and also how to advance health for all. So I'm wondering for Princess and Jorge, if you could speak a little bit about your work in terms of differences across populations.
So it was interesting because we were looking first in my case, it was looking for any discrepancy in cardiometabolic effects first. But then when we were searching the data, we found that the difference between the disparity between races, and it was something interesting that the result that we found in the therapeutic inertia and the difference between the Black and white patients, and also that there was no significant effect in the sex base when we stratified by sex base.
I don't think I could hear you, Princess. Yeah, I can hear you now.
Okay. Well, so this present study kind of ties with my previous study, and my previous study was checking the prevalence of depression among individuals with type 2 diabetes in the United States using All of Us research. And that was my first time of using the All of Us research, where I was trying to see if there was a prevalence in depression amongst individuals with type 2 diabetes in the United States. And also the most, kind of common population with that is the African American population from that previous research. And I wanted to take it, a further step into checking if this would the use of antidepressants is going to cause more metabolic outcomes for these individuals in the races. Although in this present analysis, I did not look directly into specific races or specific antidepressant use that would cause it. I just wanted to see if there was an association with antidepressant use with poor glycemic control. So hopefully in the future, I can check to see if there's an association with poor glycemic control in the use of specific antidepressants and how it affects races and hopefully genders.
Thank you, Princess. Chris, we have a question for you. Did you explore which features contributed the most towards predicting liver fat?
Yeah, that's a great question. We did look at feature importance. So for some background, this is all a tree ensemble model. So this is XGBoost. And we did look at feature importance. I do have that plot somewhere, and I would love to share that with you if you can send me your email address, perhaps, Kaushik. I think there's also a preprint that I can perhaps point you to that addresses this question. I would have loved to do that in both All of Us and UK Biobank. Of course, we only trained this on the UK Biobank side of things. But yeah, I'm happy to share that with you. I see that you've asked several other follow-up questions as well about gene validation and finding other genes. I'm not sure if you have time to address those. I would love to, but
yeah, if you can address those in maybe 1 minute or less, that would be
I will, yeah.
great.
So for your gene validation question, actually, like, big thanks to the wet lab team here. So there's like a wet lab follow-up here that actually showed that knockdown of IRS1 interrupts de novo lipogenesis. So basically, there's mice studies on this front. And did you find any other genes in IRS1 in All of Us or UK Biobank? Yes, we did. We found many more. And those are also listed in a preprint that I can hopefully share with you.
Thanks, Chris. I just want to take time to thank all of our panelists today for sharing their work and demonstrating the potential for All of Us research program's dataset. And thank you to all of you who attended today. We really appreciate your questions and your interest. As a friendly reminder, please explore scientific abstracts and posters, and don't forget to cast your vote for the People's Choice Award. There will be a short survey following this session. After completing the survey, you'll be automatically redirected to the keynote session. So thank you all for your time today. And again, thank you to all the presenters. Your work continues to inspire us all.